Diffuse. Echoes of eternity, unraveling the symphony of life. On a spring day in 2003, headlines blared across the globe. Humanity had charted the vast expanse of its genetic blueprint. The triumph was hailed as monumental, a pinnacle of scientific endeavor. Yet beneath the fanfare lurked an inconvenient truth. Vast swathes of this map remained shrouded in shadow. Far from a finished masterpiece, the canvas of the human genome bore gaping voids, overlooked not out of negligence, but due to the limits of the tools at hand. The scientific community, armed with a 13-year billion-dollar effort, had catalogued roughly 3 billion molecular notes, DNA's base pairs, that compose the human score. But the missing sections, repetitive echoes dismissed as genetic clutter, were deemed inconsequential by the prevailing wisdom. Once mocked as evolutionary debris, these regions, sometimes called the silent strings of our biological orchestra, were thought to be mere vestiges, relics of ancient viral stowaways woven into our code eons ago. The prevailing belief held that the essence of our humanity had been captured, a foundation solid enough to propel discovery forward. Yet, estimates suggest this overlooked terrain could span up to 2/3 of the genome. Even within the charted realms, pockets remain undeciphered, mysteries that whisper secrets about longevity and decay. In the decades since, revelations have upended old assumptions. Within the spiraling strands of DNA lie vital sequences once invisible to early scans that orchestrate the proteins shaping our existence. Some genes, far shorter than the thresholds once set, have emerged as conductors of life's intricate rhythms, defying the notion that the uncharted was unimportant. This journey toward a fuller map has illuminated much. Yet one enigma persists. No single switch governs the relentless march of time. Genes that bolster resilience or temper aging's toll have surfaced, offering pathways to extend vitality through nature's own mechanisms or human ingenuity. But unlike the clear culprits behind diseases like cancer, aging lacks a solitary mastermind. It's not a flaw evolution engineered into us. It's a consequence of systems designed for survival, not eternity. The humble herald of longevity. The roots of understanding aging's cadence stretch back to an unsung visionary whose legacy resonates in labs worldwide. Decades ago, a gentle pioneer named Elias Carver transformed a modest organism, a single-celled yeast, with a penchant for sweetness, into a cornerstone of discovery. Known for his generosity as much as his brilliance, Carver amassed a treasury of yeast variants at a bustling research hub, sharing them freely with scholars rich and poor alike. His altruism seeded a revolution. In the mid-20th century, yeast was little more than a baker's tool, its potential as a mirror to human complexity dismissed. Carver and his collaborator, Miriam Tate, saw beyond this skepticism. They recognized that these tiny fungal cells, despite their simplicity, shared a striking kinship with us. Complex in their own right, their genetic threads weaving patterns akin to ours. Their work revealed that yeast could unlock secrets of lifespan, disease, and resilience, earning accolades that echoed through Nobel halls. One pivotal insight from 1959 showed that yeast cells, like families, pass down legacies of longevity or brevity. A mother cell's lifespan could diverge dramatically from her daughter's, hinting at forces beyond mere genetics. By the time Carver's light dimmed in 2007, a global chorus of thousands sang the praises of yeast as a model for life's limits, a testament to his quiet audacity. Humans and yeast diverge by 1 billion years, yet their shared notes, nearly 3/4 of our genes, strike a familiar chord. Both crave sustenance and replication, their cycles slowing and swelling with age. Where humans measure decline in decades, yeast compresses it into days, offering a swift lens on time's toll. This humble organism, redolent of rising dough, became a beacon for probing the mysteries of existence. The symphony's hidden score picture the genome as a vast orchestra pit, each gene a musician poised with an instrument. Roughly 20,000 players occupy this ensemble, their strings and keys tuned by nature's hand. Alone, they hum distinct tones, but together, under the baton of the epigenome, they weave symphonies, jazz riffs of vitality, slow waltzes of growth, or dirges of decline. The epigenome, a masterful conductor, doesn't alter the notes themselves, but dictates their volume and tempo. It wraps DNA in protein coils, silencing some genes while amplifying others with chemical markers, tags of carbon and oxygen that signal when to play or pause. This orchestration ensures a liver cell strums a different tune than a neuron, despite sharing the same score. Yet this conductor is fallible. Imagine a maestro distracted. Mid-performance, missing cues as the music falters. In life, assaults like radiation or chemical breaches fracture DNA, summoning repair crews, sirtuins among them, to mend the breaks. These guardians abandon their posts, leaving genes to sound out of turn. Over time, this discord, epigenetic noise, muddies the melody, turning harmony into chaos. This theory, bold in its sweep, posits that aging stems not from genetic mutation, but from this unraveling harmony. It's a testable claim, rooted in simplicity, yet vast in scope, seeking to unify the cacophony of aging's signs, fading stamina, creaking joints, dimming faculties, into a single narrative. Lessons from a tiny stage in yeast, the drama, unfolds vividly. Normally, these cells hum a fertile tune, their mating genes muted by sirtuins. But as DNA frays, twisting into rogue loops dubbed ERCs, these guardians rush to stabilize the wreckage, abandoning their silencing duties. The E genes blare, sterility sets in, and the curtain falls prematurely. Experiments with yeast mutants mirrored this tale. Swapping a key gene with a flawed copy halved their lifespan, not through random collapse, but a swift descent into age's hallmarks, sluggish cycles, bloated forms, and barren unions. Under the microscope, their nuclei erupted into fragmented islands, a tragic ballet of decay. Inserting these rogue loops into youthful cells accelerated the decline, confirming their role as aging's accelerants. Conversely, bolstering sirtuin reserves steadied the score, extending life by a third. These findings, etched in scientific annals by 1997, heralded a breakthrough. Aging's roots lay in genomic instability, a thread traceable across species. The mouse that rewrote time. To test this symphony's relevance beyond yeast, a new stage was set with mice, ice mice, engineered with a borrowed blade from a slime mold. This enzyme, triggered by a subtle cue, snipped DNA harmlessly, mimicking life's daily wear. The result? Within months, these rodents grayed and stooped, their vigor replaced by frailty, despite no genetic tampering. Their epigenome, jostled by these breaks, lost its grip. Genes misfired, cells drifted from their roles, and the mice aged half again as fast, proof not of mutation, but of information's erosion. This clock, ticking from birth, could be wound forward, suggesting it might also be rewound. Natures. Timeless tutors consider the bristlecone pine, its gnarled limbs defying millennia in California's highlands. Older than empires, these trees show no cellular weary. Growth, pollen, seeds unwavering across centuries. Hydras, too, shrug off senescence, their lab-kept kin thriving for decades. Even jellyfish rewind their clocks, reassembling from fragments into youthful wholes. Closer kin, like the bowhead whale, stretch lifespans past 2 centuries, their cells humming a steady tune. These outliers hint at a possibility. Aging isn't inevitable. Their secret? A tightly orchestrated epigenome resisting the noise that frays ours. The shifting terrain of existence envision life as a rolling landscape, cells as travelers, descending from a peak into valleys of purpose, skin, muscle, mind. The epigenome steers this trek, anchoring each to its destiny. Yet storms, DNA's fractures, shake this terrain, nudging travelers astray. Over decades they wander, half themselves, half other, until purpose blurs into senescence. This analog system, unlike a digital lock, bends under pressure. Evolution crafted it for survival, not permanence, leaving us vulnerable to time's drift. Whales and pines steepen their slopes, holding fast longer, but even they falter eventually. Tuning the symphony, yet hope glimmers. In labs, elderly, mice dosed with Vitality's elixir outran their years, their vessels sprouting anew as sirtuins rallied. Exercise, too, fortifies this orchestra, steadying the epigenome's hand. These interventions, natural, molecular, suggest aging's tempo can shift, its notes realigned. The question looms, should we pluck these strings? The science beckons, promising not just years, but vigor. Nature's exceptions whisper it's possible. Humanity stands at a crossroads, poised to harmonize its eternal song.